This presentation is about post-translational modifications based on the research paper on acetylation and methylation of histones and their possible role in the regulation of RNA synthesis. The experiments outlined in the research paper focus on the role of histones in nuclear function, particularly emphasizing modifications such as acetylation and methylation, changing the histone structure.
Introduction. The experiments were conducted in isolated carb-thymous nuclei in vitro. The precursors being used to later modify the histones were acetate and methionine. Additionally, the research tested the effects of puromycin on synthesis of different histone fractions, for example, arginine-rich fractions or lysine-rich fractions. The aim of the research was to provide evidence that acetylation and methylation occur after RNA synthesis, thus to prove that these changes are post-translational modifications. The hypothesis of the research assumed that modifications may affect the capability of histones to inhibit RNA synthesis in vivo, furthermore concluded that mechanisms may exist allowing inhibition and reactivation of RNA production at different loci along the chromosome, implying that post-translational modifications function as an on or off switch for RNA synthesis. The initial cause of RNA synthesis suppression is the complex formation between the histones and the DNA needed as a template, so what we know as chromatin.
Inhibition of histone synthesis by puromycin. The research examined the effect of puromycin on histone synthesis by adding the antibiotic to the incubation medium. This antibiotic is used as an inhibitor of protein synthesis, which stops nuclear protein synthesis at a concentration of 100 micrograms per milliliter already. Accordingly, the incorporation of amino acids into the histone was inhibited by adding puromycin. Considering that the action of puromycin involves interference with tRNA function, for instance the release of unfinished peptides from the ribosomal site, the research concluded that histones are made on nuclear ribosomes. Furthermore, the synthesis of different histones were compared, and it was found that histones were less susceptible to puromycin. For example, lysine-rich histones seemed more resistant, reasoning that different histones possess different levels of resistance to puromycin, but the inhibition is still definite.
Modification of histone structure. In the presence of acetyl N-group, in-thymous histone was detected. It was observed that the N-terminal groups were acetylated, which is lysine-rich histone were highly acetylated, about 73 to 80%, whereas arginine-rich fraction was 36 to 48%. It was found that the isolated nuclei are incubated in the presence of sodium acetate 2, C14 had rapid incorporation of isotopes into the histone. Figure 1 shows the comparison of acetate C14 uptake into different histone fractions. When the acetyl-labeled histone was separated by chromatography on cellulose columns, it was observed that the different types of histones differ in their rates of C14 acetate uptake, and it was found that the specific activity of arginine-rich fraction was particularly high. The finding was considered that the relatively low total acetate content of arginine-rich fraction as compared with that of lysine histone. The extensive acetate uptake into arginine-rich histones was selected to study of the effect of acetylation of histone inhibition of the RNA polymerase reaction. Figure 1 shows the comparison of acetate C14 uptake into different histone fractions. When the acetyl-labeled histone was separated by chromatography on cellulose columns, it was observed that the different types of histones differ in their rates of C14 acetate uptake, and it was found that the specific activity of arginine-rich fraction was particularly high. The finding was considered that the relatively low total acetate content of arginine-rich fraction as compared with that of lysine histone. The extensive acetate uptake into arginine-rich histones was selected to study of the effect of acetylation of histone inhibition of the RNA polymerase reaction. Methylation of histones in isolated nuclei. The incorporation of C14 methyl group into histone of isolated thymus nuclei was studied, and the effects of puromycin on this process was tested. From the study, it was found that the methylation of histone proceeds after the synthesis of primary structure of the RNA. Figure 2 shows that the concentration of puromycin, which inhibits synthesis of non-histone protein by over 90%, whereas lysine C14 uptake into histones by 73%, had no effect on methylation of the histone.
The effects of acetylated histones on RNA synthesis. In 1961, evidence was found that there is a connection between the acetylation of histones and the regulation of RNA synthesis, whereby histones inhibit RNA synthesis in the nucleus. The selective removal of the histones allows for an increased rate of mRNA synthesis. Also, work on isolated enzyme systems led to the idea that histone DNA complexes are unable to serve as primers for RNA synthesis. In the acetylated histone experiment, the histones retain the capacity to combine with DNA, implying a reversible mechanism. The acetylation of isolated histones. Acetylation involves the addition of an acetyl functional group to a biomolecule. In the case of the acetylation of histones, histone acetyltransferase acetylates the N-terminal group of lysine, which is positively charged, with the carboxyl ion of the acetate. This causes a neutralization of the lysine residue and contributes to transcriptional activation by relaxation of the chromatin structure, whereas the histone deacetylase has the reverse reaction of the Hack protein. Effects of acetylated histones on RNA polymerase reactions. Aggregate enzyme preparations from the carb thymus nuclei can incorporate nucleoside triphosphates into RNA. The reaction can be followed by the incorporation of C14 ATP into an acid-insoluble RNA-sensitive product. The effects of adding arginine-rich histones on RNA synthesis by said aggregate enzyme are shown in Figure 3. As Figure 3 shows, arginine-rich histone untreated has 60% inhibition effect on RNA polymerase activity. Because the histones have not been exposed to acetylation, therefore they continue to tightly bind the DNA, not allowing the DNA to act as a template for mRNA transcription. Whereas histones that have been acetylated show a significant decrease of about 20 to 30% less in the inhibition of RNA polymerase activity. In fact, highly acetylated histone 3 doesn't inhibit at all. In conclusion, the failure of acetylated and methylated histones to block the RNA polymerase reaction is not due to their inability to combine with DNA. The results make it evident that complexes of DNA with modified histones can be formed, but under these conditions, primer activity for RNA synthesis is not lost.